expression in E. coli of cloned DNA molecules. Here we will discuss that what are the different requirements for the expression of cloned DNA and how the expression of that target gene can be maximized. First, we will focus on the requirement for expression in E. coli. If we want to get a functional product of the cloned gene, then it must be transcribed successfully, and after transcription, next step, that is translocation, must be followed. And when the nascent polypeptide chain it is synthesized, then other steps like post-translational processing and compartmentalization, they may also be important. तो जो बेसिक रिक्वायरमेंट है कि फॉरेन जीन जो है वो एक्सप्रेस करे, इसके लिए एक जो स्ट्रॉन्ग प्रमोटर है उसका होना ज़रूरी है. So in addition to the requirement of the promoter, the 3' termination point, it is also very, very important. In addition to that, there must be a ribosome binding site on the messenger RNA. And the ribosome binding site, it is being provided by a short stretch of nucleotide bases that is present upstream of the start codon. And in most of the cases, the stop codon, it is AUG, or sometimes it may be GUG. And this sequence of nucleotide that is present upstream of the start codon, it is called as the Shine-Dalgarno sequence. It helps the attachment of AUG codon on the ribosomes. Now this Shine-Dalgarno sequence, it is having a complementary base pairing with the 3' end of the 16S ribosomal RNA. If we have a look on the composition of the ribosome, then along with different type of proteins, RNA molecules are also present, and one of the RNA molecule, it is 16S ribosomal RNA molecule. And this Shine-Dalgarno sequence, its 5' end, it is complementary to the 3' end of the 16S ribosomal RNA, so that they can bind on the ribosome around the position of AUG codon. And after binding, then the process of protein synthesis can take place. In addition to that, after the protein synthesis, different steps are also required, especially the modification of the newly synthesized polypeptide, like its glycosylation, amidation, phosphorylation, meristylation. So all these steps, they modify the nascent polypeptide chain. And keep in mind that all these modifications are the processes, they may not be going on in E. coli. If E. coli we are going to use as a host organism. And sometimes such modification, they are not required for the biological activity of the protein, but sometimes it may be essential. If we want to modify the protein by post-translational modification, then such activities can be introduced in the host organisms like the E. coli or the other microorganisms. For example, a gene for meristylation like N-merystyl transferase has been cloned in E. coli from yeast so that it can introduce the meristylation in kinase proteins. So the host organism can also be modified for such modifications. इसके अलावा अगर प्रोटीन सिंथेसाइज हो गई है तो ये ज़रूरी नहीं कि वो होस्ट ऑर्गेनिज्म के अंदर स्टेबल भी रहे, क्योंकि जब ये बनती है तो हो सकता है कि ये उसमें degrade हो जाए और या disintegrate हो जाए, so the stability of the foreign proteins in host organism, it is equally important. It has been observed that when certain incomplete messages they are translated, incomplete means that sometimes messenger RNA it is truncated, having incomplete message. So under such circumstances, incomplete translation is there, so that only short stretches of amino acids are produced with no biological activity. So the host cell proteases, they can degrade such non-functional proteins. So similarly, the foreign proteins, if they are expressed by cloning a specific DNA fragment, they may also be unstable within the host organism because they may not be properly folded, or the tertiary structure it is not formed. It has been observed that there may be different type of proteins in E. coli or the host organism that can play vital role in the proper folding of the proteins after their synthesis. And those proteins that assist in the folding of the proteins, they are called as the chaperones. So when chaperones they are involved in the proper folding of the proteins, then they are protected from the host organism proteases from degradation. And there is another problem that even if chaperones are present, they may not be suitable for the folding of foreign proteins that are expressed by the clone DNA fragments. So this may be the problem. So overall, we can say that for the expression of foreign proteins, a strong promoterase must be present, and after that, the proteins must be stable. And for the stability of the proteins, they can be expressed in the form of fusion proteins. Now, fusion proteins are those proteins that are hybrid proteins and expressed by more than one genes. When fusion proteins they are produced, then there is an advantage that the end terminal of the fusion protein, it may protect the proteins from degradation by the host proteases, and later on the native protein it can be recovered by the process of cleavage by using some specific chemicals.